There's one other stain, and I'm not sure this may not show up too well because of the lighting. It's called an annexin stain. Again, the nucleus here is stained blue. Annexin is something that is expressed on cells that are damaged or undergoing apoptosis, cells that are dying. And I'm sorry you really can't see that unless we turn the lights out. The red fluorescent color indicates that essentially all of the cells in this field are dead. Again, the blue, when we counterstain for other types of cells, these were all neutrophils. They're only cells that were alive in the field. The cancer cells, again, completely killed by this treatment process. So what we can conclude at the point that we kind of got to in the last 6 months was that we are able to get these delivered to animals. They don't appear to cause toxic effects. They can be given in an animal model and not cause the animal to have any abnormal behaviors, no side effects, no problems. The animals grow, develop, everything seems normal. We are able to get them into the cancer cells and the cancer cells in a dish and kill them in the RF field. And again, I could say at this point that it was a novel idea. Last video, John. This is a video some of you may have seen from the 60 Minutes piece, and this is the so what piece to this. We have to be able to get these gold nanoparticles delivered to the cancer. And this is an example of a gold nanoparticle here with a sort of a stylized little antibody hanging off the surface of it. Not necessarily drawn to proper scale. This really molecule is a little bit bigger than that. And again, the artists here, the guys tried to show a red blood cell, just showing how tiny this was. So imagine we gave an intravenous injection of these gold nanoparticles to the individual, and of course they'd circulate through the body. Well, our idea is we want them to circulate to the body and get into the cancer here, which, again, the production guys nicely made it into an evil black tumor there. So you've got the evil black cancer here and the nice normal red tissue, and here you've got the gold nanoparticles flowing into the evil cancer. And what we want to happen is that the evil cancer has these molecules on the surface that our antibodies bind to and recognize, and those nanoparticles are then taken in. Here comes John's radio waves, and of course the cells, and unfortunately this is not what would really happen, folks. No, we're not blowing the cells up. They're not going to pop in like nice balloons. That's not what we're trying to do. I tried to get them to change this, and again, the 60 Minutes people said, no, they have to blow up, doctor. We're sorry. Okay, it's 60 Minutes. What can I say? I was afraid they'd send Mike Wallace after me, so I said, okay, you guys win. So, in point of fact, we're not trying to blow up the cells. That's not what would really happen. We're trying to get just enough heat in there that the cells would have irreparable damage done to them, and they would die over a period of several days. That's the reality of what we're trying to achieve. So this is kind of coming to the end here, and then obviously I want to spend time answering questions. Where are we now? Here we are, summer 2008. Well, I've got a manuscript that we believe is going to be accepted this week. We've had a lot of fun playing with these gold nanoparticles. I showed you that heating rate with the carbon nanotubes. We've actually found that we can get heating rates with modified gold nanoparticles that are over 4 degrees centigrade per second. Now, what does that mean in real terms? What that means is that the heating rate is so rapid that we could probably do a treatment of a tumor for as little as 10 to 12 seconds and produce temperatures that are effective in producing killing of some of those cancer cells. We've also done some work that's called mass balance work. How many of these nanoparticles do you need to get into a cancer cell? We're finding that the heat release from our nanoparticles is so intense that we are, again, experiencing temperatures around, if you can imagine, immediately around the nanoparticle of well in excess of 350 to 400 degrees centigrade. So that amount of damage produced over a very short burst of radio frequency energy, we recognize we only need to get 25 to 30 of these nanoparticles in a cell. So again, imagine, destroying Reliant Stadium with about 30 dimes put in there and exposed to radio frequency fields, and you get an idea of the scale of what we're talking about here. We've now proved, and again, I apologize, this is a manuscript that has been submitted but not yet accepted. We have answered the so what question in vitro, that means in cells in a dish. We have taken targeting molecules, added them to our nanoparticles, bound the two together, and what we've shown is that if a cancer cell is expressing something recognized by that molecule, we get a markedly increased uptake of the gold nanoparticles. After very brief exposure, we then do the RF treatment, kill the cells completely. Conversely, we did control cells that don't have that target, and it does nothing. We've put normal cells in there that don't have that target. They don't take up the gold nanoparticles. Nothing bad happens to them. Again, indicating that we can target these in these particular cells. We just started last week the in vivo work, and this is really going to be the exciting things that are happening over the next 4 to 6 months in my lab. We are growing a number of types of human cancers in a type of a mouse that we can grow actual human tumors in. So again, we can use human antibodies, human peptides, other kinds of drugs, and we can actually target these. This will tell us whether we can actually do this in an animal, kill the tumor once again without causing side effects. So hopefully next time I come back here, unless I'm run out of town on a rail, I'll be able to share that with you. We're now going to be starting large-scale toxicity studies with these gold nanoparticles. Again, we have to jump through these hoops. We're taking this forward as a novel treatment. Even though gold is approved, these gold nanoparticles aren't. We're going to have to prove that you can give them to animals and not cause side effects either acutely or over a long period of time. And by a long period of time, I mean 1.5 to 2 years. So these are studies that are starting. We are also working on a human-sized device. And again, this is with the Rutkowskis and with John. And what do we mean? Well, here is our famous favorite cancer patient with this human-sized device. So this is sort of what it would look like. It's sort of, any of you who've ever had a CT or an MRI scan know what it's like to lay on that weird table and get moved into the scanner and moved back out. That's sort of the design that we're looking at here. John, I guess, didn't want to lay down on it, so if you don't want to lay down on it, what do? You make your wife lay down on the bed. So here's Mary Ann going, oh yeah, great, don't turn that thing on. There's actually, this is empty, there's no generator there. So, you know, I really had to give you some trouble about that, John. I mean, come on, couldn't you just lay down? So the idea would be, you know, you'd have this device here, you'd move the patient through, and again, let's say your patient had a tumor in the liver or a tumor in the pancreas or a tumor in the chest, a lung cancer. You could do a treatment of that specific tumor, or again, if you think that there are microscopic cells anywhere, you could literally move the whole patient through. And again, the rate they would have to move through is a little bit slow, but you could do an entire body treatment in about 60 to 90 seconds. Because I've just told you that these cells really only need an exposure to the radiofrequency field of about 10 to 12 seconds to produce potentially lethal injury. So what happens if we can't do this? Again, this is my favorite slide, and again, this is I once again invoke my right as a surgeon to be occasionally a little bit profane. You know, the key is I really believe in this project. I really believe this is going to work, and I do believe we're going to get to clinical trials. I already have some insider information, so I can be kind of cocky like that and say I think we can target this. If we are not successful at targeting the cancer cells, or we damage normal cells, well, this is what we're going to be in. And yes, this is actually a real ranch near Stephenville, Texas. I'm happy to say my patient actually works at this ranch. He told me about it, and I had to drive up there and take this picture just because I didn't believe it. I particularly like the lounging cow here. But we actually don't think we're going to be in deep doo-doo, as my daughter would say. We believe that this does have potential. We believe that we are going to use this as a real treatment in patients with a number of types of malignancies. And now I'm going to share with what John wanted to hear. I have visited the Regional Cancer Center, Hammett, and St. Vincent's today. I was very pleased. First of all, to all of you at all three of those sites, thank you for your graciousness and for your time today. I realize that having somebody flying from out of town and take up your busy time is always an imposition, and so I greatly appreciate your taking the time to meet with me and talk with me. I'm very pleased to tell you that, again, coming from a very large cancer center where we do massive numbers of clinical trials, we do our own trials, we do cooperative group trials. You actually have an excellent cancer care system in this community. I was very impressed by the number of cooperative group trials that your cancer center participates in, the number of protocols in not only cancer but other types of state-of-the-art medical care that you are participating in. And you are certainly seeing the types of patients we need to treat in our early protocols. So I can tell you certainly have the infrastructure, and I can tell you tonight I'm committing to Erie to include you as one of the phase 2 study centers.